Yeah, thank you. I'm very happy to be able to give this talk today. I'm Noah Knorr. I'm a PhD student in the lab of Professor Linnea Hesse, and I started my PhD in November last year. I'm doing it in the university in Hamburg in Germany, together with a collaboration with the University Medical Center in Freiburg, and I'm funded by the German Studienstiftung des Deutschen Volkes, which is the foundation for academical scholarships in Germany. My talk's going to be about the biomechanics and structural adaptations in the stems of self-supporting Marantochloa leucantha. And to start, I would, so it's important, why do we work with Marantaceae in the first place? So Marantaceae are monocotyledonous family. They show the monocot typical atactostelar vascular system. And their growth is confined by primary growth, like most other monocotyledonous families. They don't have secondary thickening. Despite all this, they do have quite high growth form diversity. There are self-supporting plants, scramblers, branch angle climbers, and herbaceous plants. And we asked ourselves, what properties enable the self-supporting growth form of Marantochloa leucantha? Being based in Germany, we don't have tropical climates, so we have to cultivate the plants we investigate in greenhouses. And this is what they can look like with a small pot, and then there's a main stem that's erecting vertically, can reach several meters in height, and then we have first branching. This branching can reach quite obtuse angles of up to 122 degrees, as seen here on the right side. And then the lateral branches fan out and they form a canopy. And it struck us how the internodes are able to support the weight of this crown up top. And so the first thing we did was cross sections. And here on the left side, you can see a cross section, a light microscopic image. The left side is left plane, and the right side it's a schematic drawing where we trace the vascular bundles. And you can see it's a quite homogeneous cross section. There's a firm structure, and in the structure, you can find several different types of vascular bundles. And they are layered consecutively, and starting from the periphery, you see first A and S bundles. The A bundles have wide metaxylem vessels in the middle, and then one wide metaxylem vessel, in this case, followed by some phloem and a enclosing by sclerenchymatic tissue. The S bundles are pure fiber bundles, so they don't have any conducting properties. They're only there for mechanical stability. And this is great because the mechanical stresses that are in the plant when a mechanical load is applied to them are most prominently in the periphery of the stem. Moving further into the center of the cross-section, you can see B, C, D, E, and F bundles. And the further into the stem you go, the less sclerenchymatic tissue there is. And they can be quite easily distinguished. For example, the E bundles over here, they don't have wide metaxylem vessels. They only have smaller protoxylem and, for example, only sclerenchymatic tissue on one side. And we asked ourselves what mechanical properties do these vascular bundle organizations do with the stem. So we performed bending experiments, to be precise, 3-point bending, where we got our samples from the internodes. They had to be a specific length, so they could be bent. And they were supported on either side by lugs. And then we put a force in the middle, perpendicular to the stem, and we applied the force downwards. And the resulting force that was measured by the sensor and the distance that was traveled by the force applicator we use that to calculate the bending stiffness, which is a measure for the resistance against bending deformation. And when looking at the graph here on the left side, on the one axis we have the bending stiffness, on the other one the relative stem length from roots. So this 0% is the basal part of the stem, and this 100% represents the topmost part of the plant we investigated. And you can see 4 different colors. The green one is the main stem, blue is lateral branches of first order, orange is second order, and yellow is third order. And you can see quite a distinctive flow of the values where the most basal part of the value is the highest, and the further up you go in the main stem, the lesser the values get. And this is mostly due to the radius. The radius plays a really big role when calculating it. It's taken to the power of 4, so you have a really high influence by the radius, and this is depicted in the bending stiffnesses. And what the plant essentially does is it has a bigger radius in the bottom, and then on the basal part of the main stem, and the further up you go, the smaller the radius gets, so the bending stiffness is reduced. The lateral branches, they scatter around a pretty soft bending stiffness. When comparing different species with each other, though, this measure isn't really that helpful because the radius plays a really big role in it, as well as the cross-section geometry. So there's another measure for that, and that is the structural modulus of elasticity. And the effect of the radius and the geometry of the cross-section essentially gets calculated out in the formula. So you have a material parameter to quantify tensile and compressive stiffness. And this shows us more about the material, as we said, and it's quite contradictory to the bending stiffness we had before, where the most basal part of the main stem has the lowest value in the bending stiffness, so the material is the softest. And the more apical you get, the stiffer the material gets. So it's a way to combat the decreasing radius of the main stem and to stay erect. And this is not only the case in the single plants that are depicted here, but in 10 individuals that we investigated, they all show the same trend. The bending stiffness gets reduced the further apical you get in the main stem, and the structural modulus of elasticity is increased from the basal part of the main stem to the apical part, and the lateral branches, they sort in between those. So I talked about comparing it to different individuals and to different growth forms. For example, here we have Flagellaria indica, which is a leaf tendril climber, and typical for this growth form or for this species is that they are self-supporting in the lowest 4 meters of the stem, and then they become climbing. And this is can be seen here as well. So you have the lower 4 meters, the structural modulus of elasticity is quite high, and then it gets reduced and stays constant the further up you go in the plant. And this, it stays constant, is very typical for climbing species. You have that as well in Dioscorea, for example, in Dioscorea bulbifera and Dioscorea zanzibarensis, which are both climbing species. And here the structural modulus of elasticity stays at one level the whole way from basal to the top. This goes as well for scrambling Marantaceae. The plant doesn't really have to be hard because it's staying on the ground, so it stays flexible from the base, from the basal part to the apical part. And when putting Marantochloa leucantha in this picture, you can see the trend that we saw before. The structural modulus of elasticity is increasing the further up you get in the plant, and then you have the lateral branches that scatter around a medium stiffness. So to sum up this first part, or to conclude this first part, you can say that the main stem has to be stiff to hold the weight of the lateral branches and the crown. And this is done by the increased radius in the basal part of the plant and the decreased radius in the more apical part of the main stem. is done for with stiffer materials, so the main stem gets stiffer the further up you go to be able to support the load. And the lateral branches stay flexible. And we did, or we are trying to, and we're gonna try to, and we tried to identify anatomical and morphological traits that can be pinpointed to those results. For example, we are looking at the cross-section of the internodes again, and looking at the number of vascular bundles and the percentage of sclerenchymatic tissue you have compared to parenchymatic tissue. And what this does between young apical cross-sections and more older basal locally or basally located cross-sections. Another point for this is to look at the cell level. So when looking at the cell density, you can see in the young apical cells here on the right side, you can see an A bundle, here's an S bundle, this is a B bundle, and same goes for all these three bundles. The sclerenchymatic tissue or the cell walls of these sclerenchymatic cells get bigger the older the cells get, or the tissue gets, and the influence of this we're gonna also investigate. As a third thing, we're gonna investigate the cell wall plasticity to really get a feeling of how the cells react on the cell wall level. And to do this, we're doing synchrotron measurements at the DESI Center in Hamburg, which is a nano-CT. And to do this, we investigate the shrinking and swelling of the sclerenchymatic fiber caps in 90% and 0% relative humidity. So it's really difficult to measure the humidity of the probe itself in those small dimensions. So we measure the relative humidity of the air surrounding it. And we've only started evaluating the results 2 weeks ago, so we only have preliminary results here. But what we're gonna get, and these are only two values, so you can't really interpret anything, but what we're gonna get is a distance to base, similar as we did for the biomechanical results before, and a volumetric shrinkage to see if there's a difference in the shrinking and swelling behavior of these cells in younger apical cells or older basal cells. Now to the second part of the talk, and this is what I'm going to focus on in my PhD about, is the branching in nodes. So nodes, they are everywhere in the plant, of course. And before, we've only looked at the internodes. Now, the focus in the nodes, when you put them in a CT scanner, you can see nodal plexus. And these nodal plexuses, they connect the vascular bundles in a structural, mechanical way and in a conducting way to be able to support the lateral branches and the main stem and the leaves that are attached to them. The pink area here is an area with increased cell density. We don't know what it does yet, but we don't know what the nodal plexus does yet anyways. So what is interesting about this is that nodal plexuses are found in at least 3 monocotyledon disorders that are the zingiberales, the commelinales, and the poales. And all of these show quite a high growth form diversity. So you have, of course, Marantochloa leucantha, which is self-supporting, but you also have epiphytic plants. You have really big self-supporting plants with quite huge stems. And then you have scrambling plants like in Maranta. And we investigated Maranta leuconeura also in micro-CT. And what can be seen is that Maranta leuconeura also has a nodal plexus, and next to the nodal plexus, there's meristematic tissue. And this enables Maranta leuconeura to develop new roots and to connect these roots to the existing vascular system when the plant grows and the nodes touch the soil. Of course, it's a scrambling plant, so this is very helpful. In Marantochloa leucantha, we couldn't find a meristematic tissue yet. We're still going to look for it, but so far none have been identified. And when looking more into the function of the nodal plexus, I'm going to turn to MR imaging. So why do we use MR imaging? This is mostly used for human scales, of human scans, and you probably know it from the medical clinic if you ever needed to go into one. But the big advantage over CT scans is that you don't really need sample preparation. You can just take your plant, chuck it in the MR scanner, and you're good to go. And after you've done your scan, you can take it out, put it back in your greenhouse, and the plant's going to behave just like the plants that never left the greenhouse, because there's no real change in the tissue. You don't have ionized radiation or anything like that. And this means that you can do scans in vivo and in situ. So you can do live bending experiments inside the MR scanner and evaluate how the vascular bundles deform and how the nodal plexus behaves in this, and hopefully be able to pinpoint the functions of the nodal plexus. And last good thing is that you have good soft tissue contrast. MR imaging uses the paramagnetic properties of protons, which are found in water. So you have a lot of water in plants or in plant tissue, and this gives a really good signal. And with this signal, you get quite good contrast. That is, in this image, the contrast isn't that high, but we have a really high resolution here for MR perspectives by 40 in 40 by 40 by 40 micrometers. However, we do still have some artifacts that need to be coped with. They can be seen here on the right, for example. There's this leaf sheath, and they arise from air bubbles. So when you have samples, tissue samples, or any objects, to be fair, in a homogeneous magnetic field, these air bubbles or these objects cause the magnetic field to dehomogenize, and thus you have a dephasing of the spins, which means you lose signal. And this signal loss can be seen here. You have small black bubbles where you don't have any signal. It's more dramatic in a cross-section where you have this big black area where you just don't have signal, and this has been a fully intact plant, so there isn't any morphological things wrong with this. And to give a lookout, I'm going to focus on functional MRI in my doctoral thesis about diffusion tensor imaging, which does exactly what it says. We get tensors for diffusion, so we can weigh the images for water diffusion and track water diffusion in tissues, and about 3D sap flow measurements, so we can measure the flow in xylem and phloem, and hopefully pinpoint the functions of the nodal plexus in these experiments. And with this, I would like to thank you for your attention. I would point especially thank you to my supervisor, who's been really supportive and enabled me to give this talk here today, and to Anaclara Visconti, who did most of the work I presented here today, but couldn't be here, unfortunately, so I took over her talk. And with this, I'm open for questions.
Dankeschön. We have now time for 2 questions. Does anyone want to ask something? Okay.
Yes.
Did the hydration of the plant have any effect? Like, I assume these were taken from a greenhouse that you did these sections?
Yes.
Now, were they watered before? Did it make any difference? Or?
Yes, it does make a difference. We try to water the plants the evening before we do the measurements, just so they're very vital. And of course, when you have plants that are in a poor state, they have less water, so you have less signal, which doesn't really help. Yeah.
Does anyone?
It's sort of a technical question, but I'm thinking, you've got these really nice cross sections and you've got these various imaging techniques. Is there an ability to combine them so that you can get sort of the anatomical sectioning at each stage across? I mean, I know you have the sections through the node, but I'm thinking about this with as far as the plexus is concerned. Like how are these able to be combined together to create an overall image?
So you mean the thin slices in light microscope image?
Right. And I guess,
Well,
or the measurements that you take from them, are you going to kind of confirm those also with the measurements of the MR?
well, yeah, you can, of course. You would have to image the MR sample before cutting it up for the typical long used method of thin sections. But yeah, of course, you can combine these methods. You can't scan the sample afterwards, of course.
Thank you very much. And now I would like to invite to the stage Regine Klassen-Bockhoff.